Okay, I want to welcome Dr. Wujin Miao of the Biochemistry Department today. He's going to talk with all of us a little bit about the research that he's doing in his group. So, Dr. Miao, I turn it over to you.
All right, so I'm very happy to talk to you, all students. So, our research is mainly focused on analytical chemistry, which is part of chemistry. So the analytical chemistry in general is basically try to study the methods of determining unknown target samples. So in particular, we are interested in using electrochemistry combined with spectroscopy techniques for sensitive and selective detections of all kinds of unknown species, including regular chemicals such as explosive materials, biomolecules such as DNA, protein, or even virus. So one technique we use quite often is electrochemistry, which is involving the electron transfer reactions. And if we have electron transfer, sometimes you have light produced. So we call this technique as here electro-generated chemiluminescence. And then we use such a technique to generate or build, fabricate different types of so-called chemical or biosensors. So if you detect chemicals, we call chemical sensors. Otherwise, biomolecules, that's biosensors. So that's the main research area. And we have also some studies on so-called renewable energies. Basically, we try to develop very cost-effective catalysts for such as fuel cells, formic acid fuel cells, or water splitting to hydrogen and oxygen, which can be used for generate electricity through a fuel cell. You want to talk more on this part?
You've got about another minute or two if you'd like to continue. Yes.
Okay. So we have, of course, we have a lot of collaborations nationally and internationally, and we the paper we publish, of course, mainly in analytic chemistry journals. So here I show you one example for cocaine detection. Basically, you have DNA sequences type electron surface, and after you have the cocaine added, the DNA will bring to the electron surface to have a enhanced signal. So that's one example. And then here's another example to show how can we build a very stable electron surface attachment so that you have a very stable signal produced. So stability, that's an issue on the surface chemistry. So this shows how stable the signal we can have. And for the virus detection, here's a paper. We collaborated with a professor in biological sciences, I think 2 years ago, on Zika virus detection, and we can detect down to one plaque virus, which is, I think, a very sensible technique. And we have more to show, but it's time limit. So here just shows if we have multiple analytes, we can use the technique detect simultaneously. For example, 2 or 3 proteins, we don't need to separate. And here just shows we can detect a typical explosive, so-called TATP, triacetone triperoxide. It's quite often used by terrorists. So we can target this molecule using electro-generated chemiluminescence. So this slide just show you we synthesize different types of catalysts for formic acid oxidation. And here just shows the result. Very effective catalytic behavior enhances the signal. And this just shows how can we use water splitting technique to take advantage of the solar energy and then electrochemical technique together to decompose water to form oxygen and then hydrogen. And later on, you can use a fuel cell to generate water again. So this is kind of cycling green chemistry for the energy production. And we have more others, but I don't think you have enough time to talk about this. So
okay. That's great. That's great. So
okay.
a couple of questions that students might have.
Yeah.
So you talked a little bit about the detection of viruses. Are you currently working on the COVID virus in any way? With
No,
detection?
I'm not. I'm not, because to detect COVID-19 virus, you have to have certain certificate to handle this, but we don't have such a capability. So
Okay.
we didn't work on. And I also thought too many people compete with something. We are not in the front, so that's not not able to compete with others. So we just decide not to do that.
Okay. Okay. So I know that you take undergraduates to start research. I know you've had a number of students that have completed their capstone experience with you, but do you have any requirements for a student who might want to join your lab early? Is there any point in their undergraduate career that you would prefer them to have been through certain courses or whatever?
Actually, I like to students join the lab earlier rather than the last semester. So we had several students join the lab. I think second year, third year, they produce a lot of good publishable results. I don't have particular requirements, but I would need students to talk to me and show me the strong interest, why you are interested in this research area, what would be the career goal. So let's just find a common interest, and then we can decide from here.
Okay. I do know that we have in the Chemistry 108 class, we have about 31 students.
Hmm.
And we do have a few that have shown an interest in materials, and we've had a few that have shown an interest in kind of forensics type projects, which
Yeah.
I think might fit very well with some of the things that you were talking about.
Exactly. So forensic science, actually, we have collaboration with professor there. So many molecules targets need to be sensitive to detect. So that's our job, actually.
Okay. Well, again, Dr. Miao, I want to thank you for taking the time to talk to our Chemistry 108 students. I'm sure that they're going to enjoy what you had to present today. And if you're okay with it, at the end of the video, I'm going to put your email address. So if any students would like to reach out to you about potential research opportunities.
Yes, yes. So thank you for take care of this important part of the course, actually. I think students will learn a lot from this experience.
Well, thank you very much for that, and I'll talk to you soon.
Okay, take care. Bye-bye.